Welcome to the Ducks Unlimited podcast, the only podcast about all things waterfowl. From hunting insights to science-based discussions about ducks, geese, and issues affecting waterfowl and wetlands conservation in North America, we bring the resource to you. The DU Podcast with your host, Dr. Mike Brazier.
And we have something new for you today. As frequent listeners of the show will know, we frequently connect with scientists, established scientists, whether they be federal researchers or university researchers, and talk with them about some of the work that they're doing and how that work informs the conservation and management of waterfowl and wetlands in North America. What we're going to be doing today and what we're going to do periodically going forward is connecting with some of the other people that are involved in this research. And that includes the graduate students. A lot of the research that is conducted across North America in this regard is actually done by, or has significant involvement by graduate students. I certainly came through that route, and we get involved in all sorts of research, and most times it's the grad students that have some role in this. And so what we want to do is connect with some of those graduate students that are actively in the process of conducting their research or otherwise planning to do the research. For a number of reasons, we want to expose our listeners to some of the new and innovative research that's actually happening. We want you all to be able to hear that from the people, from the graduate students that are actually doing this work. But then we're also going to be targeting this discussion and the ones that will come in the future to a subset of graduate students, a particular subset of graduate students. And those are recipients, those are the individuals that have received what we call our Ducks Unlimited fellowships. These are fellowships that are granted to graduate students, young professionals, waterfowl professionals that are involved in some cutting edge, very important research for waterfowl and wetlands. And there is pretty strong competition for these fellowships. They're awarded annually by Ducks Unlimited Canada. And I think there are 8 of these in total. And so we're going to be throughout the year connecting with the recipients of these awards on an annual basis. So we all can hear about them, hear about what they're up to, and to give them an opportunity to share some of the exciting work that they are doing. So with that background, today I am really excited to welcome in the very first guest, I guess, formally in this vein, and that is Cheyenne Beach, a master's student at Western Illinois University. Cheyenne, welcome to the podcast.
Hi, thanks for having me.
Cheyenne, I have to correct myself just a little bit there. We had Casey Seatash on an earlier episode where we spoke with her about some of her research. Casey is another recipient of one of these fellowships. But at that time, this idea of kind of formalizing these sessions, these individual episodes with specific recipients of these fellowships had not been established. So I want to make sure I back up and give due credit to Casey for her appearance in this regard as well. Anyway, let's move on to sort of an introduction of you. I'll briefly just here tell people that your research involves lesser Scaup, and it's a pretty neat study evaluating the effects of trematodes on lesser Scaup. And I'm going to let you save all the details for you, but just to kind of give people a bit of an idea of what we're going to talk about. Now, I want to hear from you on just some of your background, your personal background, and how you wound up at Western Illinois University.
So I grew up in central New York, very outdoorsy family, a lot of hiking and biking and hunting. And I went to a SUNY school. SUNY stands for State University of New York. I went to SUNY Morrisville. That's also in central New York. Studied natural resource conservation there. And then I've kind of bounced around the country, working a bunch of different jobs focused around wetlands. And I really honed into waterfowl a few years ago. And I think I got really hooked in with waterfowl when I was working a job in North Dakota. And the following year, I'd worked for Ducks Unlimited on a project up there. And so from then on, I've been locked in. But yeah, I've been all over the country working different jobs with invasive species, shorebirds, and waterfowl.
So this isn't your first rodeo when it comes to waterfowl, and that's not uncommon. Most of the graduate students that that end up studying something that's waterfowl specific have been introduced to this group of birds and the conservation around them through some of these summer internships. A lot of them are summer internships, but others occur throughout other parts of the year as well. And that was certainly the case with me. And it sounds like it was the case with you as well. And it's pretty neat. I would imagine you had this same realization. It's pretty neat because these being migratory birds, you have to go to different places all across the country if you want to get a really good understanding of what they're about. And it sounds like you have done some of that.
Yeah, I've worked in the breeding grounds and the wintering grounds and in the migratory, these central migratory stopover areas. So yeah, it's definitely you go to the birds, not the other way around.
Cheyenne, I want to move on here quickly to the fellowship that you received. There are 8 different fellowships that Ducks Unlimited as a family of organization awards every year. The one that you received is called the Edward D. and Sally M. Futch Graduate Fellowship, and it's awarded to someone who is doing innovative research or research in waterfowl and wetlands conservation across North America. And I think this is you're a new recipient this year, correct?
Yes. Yep.
Yeah. Occasionally we have students that receive the fellowships for multiple years, depending on the level of support they need and what the resources are that are available to us. So you're one of our new recipients this year. And so I just want to take this opportunity to acknowledge the Edward and Sally Futch family for their generous contributions that make this fellowship possible. And some of the other, I have to give credit to all the other private individuals, corporate partners that have enabled all the other fellowships. And we will get to those as we introduce the recipients and speak with the recipients of those as well. But in your case, a special thank you to the Edward and Sally Futch family for their generous support of this fellowship. There's partly a reason why I didn't introduce the full title of your research. It's number one, it's because there are a couple of words that are in there that I'm not sure that I could correctly pronounce. And so I'm going to leave that up to you. And I guess the second was I just wanted to give you the opportunity to introduce the overall title and what the work that you're doing. So tell our listeners the title of the research that you're involved in.
Yeah, so I am evaluating the body impacts or physiology impacts several different trematodes, and these are the names of the trematodes: Cyaphocaudal bushiensis and Spiridotrema globus. So yeah, that's a mouthful for both of them.
Yeah, those are the ones that I was not about to try to pronounce. That's them.
There's also a second one. So we lump Spiridotrema in at the genus level. There's also Spiridotrema pseudoglobus. Morphologically, they look extraordinarily similar, but they are different under DNA analysis. But for my project, we lump them together. So that's another mouthful.
Yeah, absolutely. So, all right. So there's a number of things that we need to unpack there just in the overall introduction of this. And so we're studying the effect of what you're actually referring to. I read your proposal leading up to this as the sublethal effects of these trematode infections on Scaup. And so, the first question is, why is this even important? So anytime we are coming up with a research question, there's something behind that. We develop the question. It's either of management concern or biological concern. And then we formulate the study in order to answer that question. But, behind it all, there's some reason why it's important. So just start out, Cheyenne, by telling us why this research is important. Why do we need to know about the sublethal effects of these trematodes on Scaup?
Absolutely. So these trematodes are exotics. They don't belong in North America, but they're here now. They were carried over in the late 1800s. So they're intermediate hosts. So parasites have crazy life cycles often. And these guys use an intermediate host. It's called the faucet snail. That's also an introduced species, doesn't belong here. And it has made its way from the St. Lawrence River across the Great Lakes system, and it's in Wisconsin and Minnesota now. And it's currently in the upper Mississippi River. Scaup, a lot of birds are affected by these trematodes. It's not just Scaup, but in the upper Mississippi River, Scaup are being heavily affected by these trematodes. Mortality is occurring on an annual basis. And then on top of that, Scaup are super important to the Mississippi flyway as a whole. Like something like 60 something percent of Scaup harvest occurs in the Mississippi flyway. So if Scaup are disproportionately affected by these guys in the Mississippi flyway, and they're very important to the flyway, we want to know what we can do to kind of halt things, or maybe do some mitigation. And really just my study is really just trying to understand what the mechanisms are inside the birds, and why they're being so disproportionately affected.
Scaup is a species. Most of the listeners are gonna know this, but Scaup is a species that's below the population goals of the North American Waterfowl Management Plan. They are one of the few waterfowl species in North America that has seen a pretty noticeable decline here over the past 20 or so years. And add to that the fact that we have lacked a really great explanation for that decline. And there are a number of hypotheses that are out there. Your research here is going to be lending some evidence to help, support or refute one of those hypotheses. And it kind of relates to the spring condition hypothesis, if I understand correctly, and that there's this idea that one of the possible drivers of this decline could be some poor physiological condition that Scaup are developing during their spring migration through the upper Midwest due to a number of factors. This possibly being one of those. I think in the past, researchers had looked at selenium. They were concerned about high selenium concentrations in Scaup on these mid-migration areas in the upper Midwest. And they thought maybe that was impairing recruitment, reproduction of Scaup. But I think we've kind of dismissed that through some collection of evidence. And then there are a few other things related to food supplies and how they may have changed in these mid-migration areas. And I think the jury is still out on that one to some extent, but then potentially adding to that is what you're describing here with these trematodes that they get from eating these faucet snails. In reading your proposal, I know you cited some instances, and I think you just referenced it a few minutes ago, of mortality that these trematodes can actually cause. How many? What do we know about the level of mortality for Scaup during spring migration caused by these trematodes? And then is that the focus of your research, or are you more interested in what we refer to as sublethal? There could be effects that are damaging to the physiology of the bird, but they not necessarily to the point of causing death. So two questions there. What do we know about the level of mortality? And then is that what you're looking at?
So first I want to correct myself. I said about 60% of Scaup are harvested in the Mississippi flyway. It's about 40 to 50%. So just to clear that up. On mortality, so I'm looking at these sublethal effects. So like you said, there's a lot of layers to why Scaup why they declined. And then on top of that, why they're not recovering. So we're trying to peel back a lot of these layers, or hopefully peel back a lot of these layers with our question. Yeah. So like I said, we're looking at sublethal. We want to know if the birds don't die, say they're up in Wisconsin, they get infected, they end up being able to keep going. What body condition are they going to be in? Are they going to be able to reproduce? Are they going to be able to raise a brood? Is that a possibility with their body condition as they're continuing on migrating? Or are they going to end up just dying off later on? And I think your second question was mortality levels.
Yeah. What do we know about the level of mortality that Scaup are facing as they're migrating through? And is the mortality itself, whatever that level is that the biggest concern, or are we thinking that maybe the bigger concern are potential sublethal effects? I realize I keep asking you multiple questions here.
No, you're fine. You're fine. So yeah, like the actual mortality, it's not majorly concerning. So I looked up the USGS, so United States Geological Survey, their Wildlife Health Center, that's up in Madison, Wisconsin. They do all sorts of things. And one of their things is kind of monitoring, this mortality that's happening. So they have a data set. It's called a Whispers data set. And that's the Wildlife Health Information Sharing Partnership Events Reporting System. All sorts of diseases are reported to this database. And for the Upper Mississippi River system, basically since the early 2000s, on an annual basis, about 7,800 are dying. So it's not major, but it is definitely these sublethal effects that are concerning.
Yeah, and we don't know very much about those, right? I think that's the nature of your particular study, where you're using captive birds to study these sublethal effects, because otherwise you have to somehow measure a trematode load in these birds on their. What we want to know is, how is the condition of the bird affected by these trematodes, and then how long does that impaired condition persist into the breeding season, as you described. So that means you got to follow these birds throughout this extended period. And if you're dealing with wild birds, that's very, very difficult, if not impossible to do. And certainly when you try to figure out, is it going to impair their recruitment, that's another entire layer of difficulty to monitor. So you're using captive birds to study this question, right?
Yeah.
And so how does that work? How are you capturing the birds, and what all is involved in that?
Yeah, so we did, we're going about it in two different ways. Already at the station I work at, Forbes Biological Station in Central Illinois, we do Scaup trapping in the spring. We trap and band Scaup. So I've taken some wild caught adults and put them in captivity, and then put them through these trials. They're pretty short-term trials. They only last 10 days. So on the first day of the trial, a subset of birds are infected with these trematodes. By day 5, the trematodes should be adults and causing harm to the birds. And then by day 10, they should be starting to die off. The trematodes, I hope not the birds, but the trematodes should be starting to die off at that point in time. So we can capture, this kind of continuum. And we're taking blood samples throughout the trial to kind of see what happens, from infection to adult stage of these trematodes, and from adult stage to when the trematodes actually die off. And then we're also, we collected some eggs up in North Dakota of lesser Scaup and hatched them out. So then we have kind of this blank slate compared to these wild caught birds that have, all of the parasites that a normal bird would have.
I don't know very much about how these trematodes affect the Scaup, but why do they die? So why do they die? And then what aspect of the physiology is affected, and why is it affected? Are these like tapeworms, where they consume the nutrients that the birds are taking in, and therefore the birds just die of malnutrition and effective starvation? Or is there some other kind of disease transmission going on?
It's definitely the trematodes themselves taking in their own nutrients. Yeah, so they work just like a lot of other intestinal parasites. These guys are, relatively small, but they're little round guys, and they just do an incredible amount of damage. Opening these birds up and looking at them after they've been infected, they have completely destroyed some sections of intestine, where other, parasites that the Scaup have evolved with, they're not causing that amount of damage. And honestly, we don't know a whole lot about the whole mechanism, this whole relationship between the birds and the parasites. These trematodes in particular, we're not really sure why they cause so much damage.
Yeah, and so your research is going to be answering questions like, I think, if I understand this correctly, like how does the different quantity of trematode, or are you studying the effect of different quantities of trematodes? Let me first ask that. Or is it a standard level of dosage?
Right now, we're going at a standard level of dosage. We went out trying to, we were going to do, various levels of dosages, and, infect over a series of days as well. So you can get at this, multiple infection and kind of replicate what would be happening as the birds are, in these upper pools of the Mississippi River, day one of getting to the Mississippi River and starting eating these snails, they're not going to be affected yet by the trematodes. There's a couple of days of delay before those first round of trematodes really start taking hold. So, they're slowly, day by day, up until about, day 5 of being there, slowly, packing on, layering on the amount of trematodes that they're exposing themselves to. But we first need to get this baseline and start seeing what's happening. So, we've kind of scaled back, and that's what I'm really trying to focus on.
Do we think some birds are able to withstand the effects of the trematodes, and then the trematodes eventually pass through the bird? Do we have any of that knowledge? Or once they consume these trematodes, are they with them, and are they just doomed? It's just a matter of time before they reach that final stage.
So far with my sublethal dosing, none of the birds have died on trial. So it's definitely, it has to be possible that, some birds are able to move on with their lives in maybe in a slightly worse body condition than when they first were exposed. Yeah. So that's definitely, potentially happening.
But that's like the core, that's like the most important aspect of your study, right? Is trying to figure out or
yes.
answering that question that I just posed to you is, what do we know about whether these birds are able to survive or what kind of body condition they're able to come out of these infections with? And then are they able to recover some body condition into the future? And I don't know how many of those questions you're going to be able to answer. The other aspect of this that I was thinking about while reading your proposal is that ideally we'd then like to have these birds be affected by the trematodes and then measure some sort of reproductive output. But I don't think you're able to do that, right? You're just kind of measuring the body condition of these different birds at different stages after infection, right?
Right. Yeah. Ideally, that would be fantastic. But my project is kind of this, we've got to start somewhere, and this is where we're starting.
Yep, absolutely. I understand. It's a really exciting project because it's one of those aspects of waterfowl ecology where there's there's a little bit of mystery around it. It's like we've talked about the mortality. We don't think that mortality that we're observing in the upper Midwest is significant enough to be contributing meaningfully to the overall Scaup decline. But then the big unknown are these sublethal effects. But it's kind of like the selenium concern that we had a few years ago. We thought that may be operating in the same way we're speculating here. Turns out that Scaup are able to recover from those selenium loads pretty quick. They're able to kind of flush those through the body. And this may be the same thing here. We just don't know until we do the research to answer that question. And you're exactly right. That's the way I was viewing this. Your study is sort of the first foray into studying in hand the effects of these trematode infections on the Scaup. It's certainly pretty exciting to hear about it.
Yeah, absolutely. And there were some studies back in the early, or I guess late 80s, early 90s, birds were being heavily affected up in the St. Lawrence River around Quebec. There was a study or a couple studies done in New Jersey, a wide array of birds that are being affected, everything from like swan to blue-winged teal. These things have been looked into briefly though. It's not, we definitely do not have a full picture of what the heck is happening.
Well, Cheyenne, we're going to start wrapping up here. I have a couple of other questions for you. The first is just how far along into your research are you? And do you have another field season, or was this the last of your field seasons?
Yeah, so I am about 2/3 of the way through, I suppose. I have a trial coming up this fall, so it'll be my last trial. And yeah, then we're wrapping up after that.
Okay. Well, I'm not going to ask you at this point to divulge any preliminary results. I'm curious of that myself, but I'm gonna I'm gonna resist the urge. We'll connect with you again, likely once all of this is complete, to get the nitty gritty of what you actually discovered, and where we may go next. Obviously, there are a lot of questions about what are the implications of this if we do find that it has some sort of persistent effect on the condition and concerning effect on the condition of these birds. Well, then what? And again, it's kind of premature to have that discussion as well, because we don't know what these potential effects are and if they are even worth being concerned about. Maybe the birds are healthy enough to withstand them, at least those that don't die. So we'll reconnect with you in the future to hear about the final results. Last question I have for you relates to the partners involved in this research. We've mentioned many times that no project, whether it be a science project or a conservation on the ground, conservation habitat work project, is done by any single entity. That's very, very rare these days. So I want to make sure that we give you an opportunity to acknowledge all of your partners and your collaborators and supporters of this research.
Yeah, so the funding comes from the Federal Aid and Wildlife Restoration Act, distributed by U.S. Fish and Wildlife Service and the Illinois Department of Natural Resources. And then, everybody that helps out on this project, all the folks at University of Illinois, Illinois Natural History Survey, obviously folks at Western Illinois University. I have folks helping me out at Lafayette College. It's in Pennsylvania. And I also have folks helping me out at University of Arkansas, Monticello. So yeah. Lots of hands. Yeah.
And of course the DU fellowship folks that we mentioned already, they're an important player in this as well. And one thing I did want to stress here is something you mentioned, that one of the key funders of this work comes from the Federal Aid and Wildlife Restoration. That's commonly known as Pittman Robertson. And that money in that fund comes from the sale of guns and ammunition and a few other things. And this is an important example of how hunters, and through their interest in this resource, their participation in these resource activities, actually help fund some of the work that is enabling us to do a better job managing the resource for the use of the current generation and future generations. So to all the hunters and archers and everyone else that contributes to the Pittman Robertson Fund through their purchase of ammunition and guns, we thank you for that. So Cheyenne, any final comments from you with regard to your research? We've enjoyed the opportunity to catch up with you and hear about this, and I hope the listeners have as well.
Yeah, thank you for having me. And thanks for the fellowship. It is fantastic. All I've got is just that Scaup are confusing. So that's all.
Well, we have to start somewhere with some of these questions. And it is. Scaup do remain one of those species that are a bit perplexing. We're learning more with each passing year. And we've come a long way to draw a parallel to pintails in understanding what drove their declines. For a number of years, pintails were much in the same boat, where we saw some declines, but we weren't exactly 100% certain what was going on, what was driving those declines. We had some hypotheses, and gradually through time, through investments in some well-crafted science, we were able to really dial in on what's causing those declines, at least what's playing a big role in those declines. And we're on that same path with Scaup. And your research is an important part of that. And we thank you for it. We thank you for your participation in that. And thank you for joining us here on the podcast, Cheyenne. It's been great.
Yeah, thanks for having me.
A special thanks to our guest on today's episode, Cheyenne Beach, a master's student at Western Illinois University. We certainly appreciate her work on helping us understand more about Scaup and what could possibly be explaining some of their decline. We look forward to the results that she gets from this research. As always, we thank our producer, Clay Baird, for the great work he does editing these podcasts and getting them out to you, our listeners. And of course, you, the listeners, we thank you for joining us here on the podcast, spending your time with us. And we also thank you very much for your support, passion, and commitment to wetlands and waterfowl conservation.
Thank you for listening to this episode of the DU Podcast. Be sure to rate, review, and subscribe to the show and visit www.ducks.org/DU Podcast for resources based on today's topics, as well as access to more episodes. Opinions expressed by guests do not necessarily reflect those of Ducks Unlimited. Until next time, stay tuned to the Ducks.